Good morning. In this part of the video of enzymes, we'll be talking about the portion of enzymes covered in A levels and MCAT examinations. So now, what are cofactors? Cofactors are the non-protein part of the enzyme, which are required for its proper functioning. So what is the function of the cofactor? And what the cofactor do is basically that they form a sort of a bridge and helps to provide some of the chemical energy which is sort of the activation energy which is required for the reaction to take place. Now the cofactor, as we all have understood, that it is required for the proper functioning of the enzyme, and it exists in two main states: inorganic states or organic states. In inorganic states, it mainly exists in the form of ions of metal atoms, such as iron, magnesium, and copper, as depicted over here. Now, in this ion form, in the inorganic form, it is known as an activator. However, if it is formed in the organic form, it is referred to as coenzymes or prosthetic groups. Now, mainly, when they exist in the form of a coenzyme, the cofactor is mainly loosely attached to the protein part, and the coenzymes are mainly made up of vitamins. The main coenzymes, which examples you need to know, are ATP, NAD, and FAD. NAD and FAD are have been also studied in the glycolysis and aerobic respiration pathway. So if we've studied that a cofactor is covalently bounded, I repeat, covalently bounded to the protein part, then it is known as a prosthetic group. The prosthetic group has a permanent attachment whenever the bonding between the two parts is covalent. It is always a permanent attachment, unlike coenzyme formation. Coenzyme formation is a reversible process. However, when a cofactor is bound, it is always irreversible. So now you need to know that whenever basically an activated enzyme consists of the cofactor within it, it is referred to as a hollow enzyme. And although the word hollow implies that it has nothing within it, however, you need to know that the hollow enzyme refers to that part of that enzyme which has been activated due to the presence of cofactor, whereas that enzyme which has the coenzyme or prosthetic group removed from it, it is referred to an apoenzyme. So apoenzyme is an incomplete enzyme, whereas holoenzyme is a complete enzyme. Here are some of further, as we discussed above as well, that majority of the vitamins are labeled as coenzymes. Here we can see vitamin B2, B3, and vitamin B12. Vitamin B12 is also present in red blood cells, and a deficiency of basically because basically vitamin B12 and red blood cells, they are required synergistically for transport in the large intestine. Without vitamin B12, a person would develop a megaloblastic anemia. Now comes the second part of enzymes, which we did not study previously. That is the inhibition of enzymes. So that you need to know that there are two types of enzymes. I'll try to just navigate over the part. So as I was telling you, I wanted to basically just get a hold of the text as well so you can see while I'm talking. So now there are two types of inhibition: competitive inhibition and non-competitive inhibition. These can be further classified into two major categories: reversible inhibition and irreversible inhibition. Majorly, competitive inhibition is referred to as reversible inhibition, whereas the non-competitive inhibition is always in the mostly in the category of irreversible inhibition. What happens is that, as we know, that in the process of how an enzyme works is that a enzyme has a lock or which is the active site on it, and the enzyme has a specific key or substrate for it. However, if and in the case of competitive inhibition, a substance which resembles the shape of the substrate attaches to the active site. What happens is that when the actual substrate or the active substrate will try to attach to the active site, it will no longer be able to attach to it because the structure which had resembled it has attached to it. Now, how can competitive inhibition, this kind of inhibition can be overcome? This can be overcome by increasing the amount of the original substrate. By increasing the amount of original substrate, the original substrate will bind to the active site and it will remove the the inhibitor which has binded to the active site. Now, in contrast to it is non-competitive or irreversible inhibition. In this type of inhibition, what the inhibitor does is that it binds to either the active site or to any structure outside the active site. When it binds to any structure besides the active site, that is referred to as allosteric inhibition. Okay, you need to remember that name. So now, whenever it binds to either to the active site or any structure besides the active site, what it does is it basically destroys the enzyme and it changes the structure of the active site in such a way that whenever even when the actual substrate tries to bind to the active site, it cannot, because now the change has shaped. So no matter how much you increase the quantity of the substrate, it cannot bind to, and this form of inhibition is referred to as irreversible inhibition. Now I would like to tell you the main examples of reversible and irreversible inhibition. You would have known that cyanide is used as a very fast poison in cases of suicide. This is because it acts as an irreversible inhibitor. And similar is the case with many of the famous poisons which are used in modern day. Here, in over here, they have explained how cyanide basically inhibits cytochrome oxidase, which is required for cellular respiration, and this acts as an irreversible inhibitor. Now, as I talked previously as well, they have shown by figures how competitive inhibition occurs, and in this example, they have the example for the syllabus is that how malonic acid and succinic acid competitively inhibit each other. So this wraps up the class main and the chapter of enzymes. And now in the classification of enzymes, this is just a bit of a memorization. However, you can know, you'll just have to read them once. That oxidoreductase, just by the name, as the name suggests, that they are involved in both oxidation and reduction reactions. Similar the case with transferases, in which they help to catalyze the transfer of a functional group. Now come hydrolysis. Hydrolysis help to catalyze the breakdown of large organic molecules into smaller ones by adding water. And in the case of lyases help to catalyze the breakdown of specific covalent bonds without hydrolysis. Without hydrolysis means without the involvement of water. And ligases, as we studied previously, that ligases are like glues. So, like, what do glues do? Glues tend to join substances. So, ligases are DNA and RNA polymerases, and they help to synthesize DNA and RNA respectively. So, this wraps up our video on enzymes.